Satellites, only thing you have to know is this: satellite nucleic acids, the gene for one, the code of one is in the main one. And satellite viruses, they're two independent viruses. They're always associated with each other. They're almost always RNA. Diseases of crop plants, bamboo, chicory, grapevine, all that. And there's only one known animal satellite, and it's a bee satellite, as in bee, not the capital letter. All right, let's get to the stuff that's fun for today. Spongiform Encephalopathy. Oh, golly bum, I love Spongiform Encephalopathy. Spongiform Encephalopathy. So let's take the word apart and see what it is. Spongiform Encephalopathies. First of all, spongiform, what's that mean?
Holes?
Right. Holes like a sponge, like a natural sponge has holes in it, or like Swiss cheese. That's spongiform. And encephalopathy, pathy means disease of. Encephalo means brain. So it's a brain disease where you get holes in your brain. Not pretty. Okay? And spongiform encephalopathies come in these varieties. Sporadic spongiform encephalopathy. Every animal has been found to naturally get this by mutation. Now, what causes the holes? All right, a normal brain protein that makes up the neurons of the brain, called PRP, comes in two forms. And it's the same length, the same amino acids with a mutation in just one spot, 1 to 3 spots, causes the protein to fold a different way. The normal one is PRP. The misfolded one is called a prion, or prion. The one that has a mutation in just one spot, I think it's 284 nucleotides long, DNA bases, causes it to form a stable, indestructible shape. That's the disease one. The normal one, shaped like this, and it works. If a normal one comes across an abnormal one, it converts it like a magnet, and they stick together in long chains, and they're non-functional, so the cell dies. So if a normal brain protein is exposed or touches, physically comes in contact with an abnormal prion, it will convert it, refold it to the stable shape that's non-functional, and these non-functional shapes will stick together in long chains, and since they're non-functional, the cell will die. And when the cell dies, it leaves a hole behind. And so this is called Spongiform Encephalopathy. What's sporadic? Every animal we have ever known and investigated will get a mutation, 1 out of every gazillion, and that animal will get the Spongiform Encephalopathy, and that animal will die, and that's it. So these are sporadic, random encephalopathies that happen just rarely, and in humans, it's been given a name, Creutzfeldt-Jakob's disease, CJD. It is the sporadic, random, mutational form of this in humans. And it's extremely rare. It happens something like 1 in every 500,000 every 50 years. So there is very rare. It doesn't seem to be related to anything except age. The random, sporadic seems to occur in the 50s and later. And the first symptoms are very much like Alzheimer's, but it quickly gets so much worse. They have hallucinations. They have random events in their brain that makes them, if there's, like for instance, you show them a movie that someone's being tortured, they feel like they're being tortured kind of thing. So it's, that's the sporadic, random one, and it's very rare. All right, then there is genetic spongiform encephalopathy in people, and there are 3 of them: fatal fetal insomnia, Gertsch-Straussler-Schlinkner disease, and Kuru, which occurred in New Guinea, probably as a random originally, many generations ago. Now, what do I mean by this? A genetic inherited trait causes the mutation that causes the baby to be born with a spongiform encephalopathy, and the baby soon dies. Fatal fetal insomnia, the baby can never sleep. So the babies die very quickly. Since they don't reproduce, they don't pass it on. So you don't really have to worry too much about the genetic form or the sporadic, because you would have already had it or developed it later. What you have to worry about is this one: transmissible spongiform encephalopathy. We didn't know that spongiform encephalopathies could be contagious until the 80s. Before the 80s, we only saw it in sheep and goats, and it was no other species got it, so we just thought, well, this is too bad for them. Now we know that it is contagious, and that's called transmissible. And then finally, there is idiopathic, and that means we know the person has spongiform encephalopathy, we just don't know how they got it. Was it contagious? Was it genetic? Was it a random mutational event? We just haven't figured it out yet. One of them is you can get it from medical instruments. Remember, it's indestructible. So if you use a medical instrument on someone that has spongiform encephalopathy, it can't be sterilized, because nothing will destroy this except for 60 hours of burning in a fire of extreme temperature, or soaking in sodium hydroxide for 6 hours, plus autoclaving at a higher temperature for a longer amount of time. So basically, nothing will destroy them, and it's almost impossible to sterilize. Let's see, and that's, by the way, if it's caused by medical instruments, that is a type of transmissible, and it's called iatrogenic. And several people have been operated on with scalpels or respiratory equipment that was used on someone that had it, and they got it. So they are now, if you find out someone has it, of course, all those instruments must be destroyed. All right, so let's talk about the history of spongiform encephalopathy. And we're going to go down here, and we're going to talk when we first heard about it. And we first heard about it in 1732. In 1732 in England, a book was written about sheep going crazy. Now, it's very hard to tell about crazy sheep because they're stupid. You know, when they say you follow them just like a sheep to slaughter. All right? Sheep will see other sheep being shot and killed, yet they'll follow. So sheep are incredibly dumb, stinky animals. I hate working with them. But anyway, in England, which sheep is about 1/3 of the meat diet is sheep in England, over many years they saw in the early 1700s the first development of this disease in sheep. And what the sheep did was they acted more crazy than dumb sheep. For instance, they will, if rain hits their head, they will look up, their mouth will fall open, and they will drown in a rainstorm. They will walk, they were thirsty, and they will walk to a stream, and they will walk under the water and forget to stop and drown. They will be curious and walk off a cliff. But the way it got its name is they have random itching. They itch. Every square centimeter of their body itches, so they rub every bit of their fur and skin off until they're bloody from head to toe. which is called scrapie. That's why they named it. Now, in the history of scrapie, no one has ever gotten scrapie from eating a sheep that had scrapie. In fact, in the old days, when they saw a sheep that had scrapie, the landowner would give it to the peasants and say, make soup out of it quick before it wastes away to nothing. No harm, no problem. What's the cure for scrapie? Same as viroid diseases. If your neighbors find out you have a scrapie sheep, they will kindly come visit you with shotguns and kill all your sheep, burn the bodies, and bury them. So it is in the farmer's interest that if they see a sick sheep, to quickly get rid of it or send it away to slaughterhouse. Because if their neighbors find out, they lose their entire farm. So anyway, story of scrapie is, that's it. We didn't expect anything more than that. No other species got it. Nobody got it from eating it. It was limited to sheep. Probably started out as a random mutation, and they were probably nosing around the dead body, and that's how it spread in a herd or whatever. So that's scrapie. The next time we saw it was in the early 1900s, repeated in the 1940s, in a really strange place called Papua New Guinea. If you look at a map of the world back there, you will see above Australia a big long island called New Guinea. Half of it is a free country called Papua New Guinea. The other half is forcibly occupied by Indonesia, because when the Dutch left, the Indonesians took what they had. And anyway, in 1900, missionaries reported that a certain people called the Faro people, or Faro people, in New Guinea, women and children had this wasting syndrome. And they died quickly and nobody knew what it was. During World War II, New Guinea was a part of, remember, the Allies made a kind of wall of ships and forts around Australia and New Zealand. And so New Zealand was a place for Allied forces. And the Allied forces reported again this disease, which was called kuru by the local people, that means to shake in fear. After World War II, a man by the name of Karl Gaidushek from Harvard went there because he thought it was a nutritional deficiency disease. He thought it was a missing amino acid, vitamin, or mineral. He got there, he did these tests, he found it wasn't. He then autopsied some of the bodies and sent it for, investigation. Didn't find a new virus, didn't find a new bacteria, couldn't relate it at all to anything. Finally, to figure this out, he went to live with the people and discovered they were ritual cannibals. When someone in the village that was important or well-loved died, they had a barbecue and ate them. And the logic behind it is, if somebody's important to the village, you want to keep their spirit in the village. And why did men and women differ, and children, in who got it? Because not many men got it, but mostly women and children. And the answer is where it's located. It's a brain disease. A disease of the spinal column, spinal cord, and brain. As a result, who gets the best parts when you have barbecue? The men do. And the women and children are left with the icky stuff, the organs and whatever, and brain and spinal cord. So he got a Nobel Prize for discovering this disease was caused by ritual cannibalism, and with efforts to re-educate and so forth, this no longer is being done, but people are still coming down with it there today because it has a long survival. Remember, they're almost indestructible particles. There's a lot of little videos online that are very interesting, especially one where they interviewed the last living cannibal, and she tells you how delicious it was and what parts were the best. but you can Google it and see about kuru. Interesting, kuru probably started as a sporadic Spongiform encephalopathy, but of course when you eat someone that has it, it spreads within a species. All right, so when was the next time we saw it? The next time we saw it was in the late 70s in England. And here's what happened. England joined the European Common Market, now called the European Union. When you join the European Union, you have to make your laws agree with theirs. And cattle feed had a lower percentage requirement, legal requirement in England than it did in Europe. And so the rest of Europe's, England's law had to be brought up to the standard of Europe. And so if you know anything about the European Union, they have laws for everything. I mean, California is considered the most legalistic state in the Union. We have more laws than any other state. But the European Union has 10 times more than we do. They have a law on the size a bus bench must be. So, they have a lot of legal requirements, and one of them was that the cattle feed had to be upped in protein percent. So if you're a company that's making cattle feed, and you need to increase your protein, are you going to go out and buy something expensive like soybeans to increase the protein of your feed? Or are you going to get the cheapest protein you can find? Cheapest you can find. And so what the companies in England did was, they went and bought what we call renderings. And you can start kind of looking in these terms. And renderings are parts of grown animals that, when they're sick, they're sent to the slaughterhouse, and usually sick animals are not slaughtered for food, they're slaughtered for cat food and other things. And if you have a part of an animal that can't be eaten, that goes into renderings. And renderings are dried and then pelleted and sold as protein. So gristle and a little bit of spinal column and spinal stuff and a little bit of lower brain is all in renderings. And remember, in England, what's 1/3 of the food meat that you eat? Sheep. So when a farmer would see a sick sheep, instead they would immediately send it to get some profit from the slaughterhouse. And it would go into renderings. So remember, if you're thinking about a cow, they make the horns and the hooves into gelatin. They make the tail into oxtail soup. They take the hide and make shoes and belts. They take the fur and they stuff pillows with it. They take the bones and grind it up for calcium supplements. They cut the steaks out. They cut the organs. You have liver, you have kidney sold. They take the intestines and wash them out and stuff them, and that's what we call sausages. They take the hoo-hoo, the hee-hee, the nose, and they grind it all up and add spice, and they call it potted meat. They take the ears and dry them and give them as dog treats. So you have just about every bit of the animal is utilized in some way, except for gristle and spinal cord and lower brain. They'll even take the brain out. My mother used to eat scrambled eggs and brain. Cow's brain. Oh my God. Anyway. So, what happened? Well, what happened was that cows got their own Spongiform Encephalopathy transmissibly from eating scrapy sheep and other sick cows that were in renderings. And first 10 cows and then 100,000 started, and they go crazy, they can't stand up, and cows are very smart animals, and when they get really aggressive and go crazy and can't stand up, it's very different from their normal behavior. And so this is called BSE, bovine Spongiform Encephalopathy. But almost everything eats cows. So then cats and dogs started getting Spongiform Encephalopathy, then zoo animals, and finally people that were teenagers and young adults started getting their own Spongiform Encephalopathy. By the way, the human form that you get from eating a cow that has it, BSE, is called CJD-NV, new variant, CJD-NV. Sometimes you'll see little v, CJD. What's the difference? If you get CJD naturally and sporadically and randomly, about 40% of your brain is holes. If you get it from eating a BSE cow, it's 60% of your brain goes to holes. So there is a little bit difference in the autopsy results of whether you have the new variant kind or the others. What's the big difference? The big difference is species barrier. You don't get canine distemper because you're not a dog. It's a species-specific disease. This is also, but there are different levels of species barrier. The species barrier between sheep and cows is low. The species barrier between sheep and people is very high. We don't get their diseases, they don't get ours. But sheep and cows can get the same diseases or similar diseases because they have a low species barrier between them. And the saddest thing is, the species barrier between cows and humans is almost non-existent. In fact, we get almost everything they get. That's one of the reasons we didn't used to drink fresh milk, because of so many things you can get from cows. So the species barrier from cows to other animals is quite low. So we had an intermediary. We had scrapie go to cows, and then cows, whoever ate them, it went everywhere. So what happened? Well, England, first, they resisted it. Nobody could believe this disease had moved into cows and humans. They finally accepted it, and they killed every cow that had ever eaten any of this food. So they killed millions of cows. Then they developed a quick test. So if you go to England and eat meat today, you are safer than anywhere else in the world because every animal that is slaughtered is tested before the meat is sold. Nowhere else in the world does that happen. In the United States, we only check 1 out of every 250,000 slaughtered randomly, and we check all sick animals. We used to say, and the movie will tell you, that we have no BSE in the United States. We have now had about 50 cases, and we have not had any people come down with it from eating animals they ate here. But we did, the first 2 cases were calves that had been fed rendered remains in Canada. They were sold to fatten up in Washington State. They were fattened up and sold for food. And excuse me, the testing is so late here. We don't have an instant test like the British do. That by the time they found these 2 animals had BSE, the meat had already been sold at Vons and Ralph's throughout the West Coast. So even though no one has ever been infected with it here, we have a pretty strange system because our law says that if any country in the world has BSE, they cannot export meat to the United States. But if someone bans our beef because we have it, we have a trade war with them. So Korea banned our beef because we had BSE, and we then retaliated against them and made them buy our beef. So it's a little bit of hypocrisy there. Well, how else can you get CJD-NV? Well, the way in which people got it in the United States was not from eating BSE cows, but hunters eating deer and elk that they shot in the western United States. 30% of the wild deer in Colorado have their own form of a Spongiform Encephalopathy that can be passed to humans, and that's called chronic wasting disease. And good news, you can go to a sports store and buy a dip test. So if you shoot a deer in the western United States and you want to know if it's got BSE, you can dip this into the fluid around the brain and it will tell you. Another interesting thing is, of course, they just tell you to cut the steaks out now from wild deer that you shoot. Don't eat the whole animal, don't cut into the brain or spinal cord, just cut out the steaks so that you can harvest the animal in a way that won't contaminate your food source. So about 25 hunters in the United States have gotten CJD-NV from eating wild deer that they shot and harvested either improperly or didn't have a dip test for. All right, so that's all we know about it. If you would open your lab record book and try to copy these little things. I don't know why this is showing a little bit of difficulty here. Let's see if I get it from the other website. There's the terms. So put one of these on each line. Protein-only hypothesis. And remember, for years and years, nobody believed any disease could be caused by something that doesn't have its own RNA or DNA, and nothing could replicate without it. Now, this is not actually replication, remember, it's conversion of a normal to an abnormal shape. And the man who came up with the hypothesis is Stanley Prusner of the University of California, Berkeley, the very best university in the world with more Nobel Prizes than any other. So he came up with the protein-only hypothesis, and he is the one that coined the term prion or prion. Scrapie, Carl Gajduszek, who got the first Nobel Prize. By the way, Stanley Prusner got a Nobel Prize also. Creutzfeldt-Jakob's disease, that's our form, the random. J.S. Griffith was the person who, a mathematician, who first came up with an idea that a protein could refold another one. The normal brain protein PRP, renderings we discussed. Cattle that fall down because they're sick are called downer cattle. Cattle with Spongiform encephalopathy is called bovine spongiform encephalopathy, and the common name is mad cow disease. If you get it from eating another animal or in some way from like contaminated instruments, it's called transmissible encephalopathy. The weird shape is called a rogue PRP or prion. When we eat a cow and get it, it used to be called human BSE. Now it's called CJD-NV. So these are the little terms that you should copy down, one per line, or skip a line between each if possible, and take notes from the video that you're going to see in approximately 5 minutes. I wish I could make this bigger, but I just don't know how on my screen.
Let's see if there's a control and then scroll up.
Like control one, like this. Oh, did it? Yeah, it did. Thank you. You're a whiz. That helps, doesn't it?
Yes.
See if I need a bit more. Yeah. Yep. There. That's the max. Because I have brain disease myself. Huh? Oh, my brain disease? Oh, yeah. Actually, he said carry a notebook. Control, roll up, right. ready yet? What do you mean, no? Who remembers what color of exam they had yesterday or whenever we took it? Okay, I've got some extras. I mean, I'm gonna put it online, but I just thought I'd hand them out. White? Here you go. Blue? Second round? You had a white. You had a blue. Sorry, I'm just sort of passing them out a little bit to try to share. And blue. What? You guys even remember?
Blue? I think it was.
Share. One blue. And the white. You said blue, right? Okay, that's all. You can share, but it'll also be online.
Like maybe.
Everybody done? Say yes. Need some sleep. Yeah, did you hear that report today?
No, is that a report today? I'm just a genius.
It was an amazing thing that they did this, WHO did a study of memory problems versus grades on math tests. And they found that lack of sleep causes a 20% reduction in your test score. And a country in the world that had the most kids with a lack of sleep, USA.
No, but I did hear on the radio this morning that in Japan that there's been a bird flu occurrence.
Another? Ooh, from China, huh? So far I've been limited there. So sad to see this video. I know you're supposed to feel sorry for people more than animals, but I worked with cows for so long, and they're such wonderful animals. They're huge, but they're just like dogs, you know. You name them, they will do things, and it's just, they will actually, the oldest, meanest female runs the field. It's amazing. Cows are just amazing animals, and they're so incredibly smart. And here you are working with this 1-ton animal, and it's like it's probably smarter than a dog, or it's just amazing. And to see them just go brainless.
What are we talking about?
City slickers.
Yes.
I was like, everyone's like, what?
All right, here we go.
A silent killer attacks the brain and claims its first human victims.
He started hallucinating.
He had very jerky movements. I think we all had to agree that this was something unexplainable.
Past outbreaks offer chilling clues of how it spreads.
It was clearly being transmitted from person to person by cannibalism.
Is mad cow disease a threat?
These agents are almost immortal.
the brain eater.
And people like you. For my comfort.
It appeared suddenly in England, a strange and horrific plague called mad cow disease. Almost overnight, cattle throughout the country became infected, shaking uncontrollably, turning aggressive, and losing coordination. There is no cure for this mysterious disease, and it is always fatal. Desperate to halt the spread of mad cow disease, England destroyed nearly 2 million cattle, but it was too late. Since 1985, thousands of infected cows may have entered the food chain. It now seems certain that mad cow disease is crossing into a new species. Recently, an 18-year-old named Stephen Churchill fell ill with some very curious symptoms.
He had very jerky movements, and he could suddenly decide to try and get up and walk when he wasn't capable of doing any kind of action, which would often result in him injuring himself or falling over if there was nobody around to help him.
He started hallucinating. It started off as he'd be watching television and he'd get very enthralled in what was going on. If there was fire on the television, he'd feel as though he was burning, or if it was like an underwater scene, he'd feel as though he was drowning. And then it got to a stage where he was just seeing things that just weren't there.
At the time, the cause of Stephen's illness was a mystery.
We saw CJD and a question mark written on the notes, and that was the first sign of they had maybe an idea of what it was. I found out that it was a very strange disease, but I also found out it was only really found in 50, 60, 70-year-olds. And although a lot of the symptoms seemed to fit, the age group didn't.
Doctors thought that Stephen Churchill might have CJD, or Creutzfeldt-Jakob disease, a rare brain ailment in the same family as mad cow disease. Called Spongiform Encephalopathies, spongy brain disease, these illnesses riddle their victims' brains with holes. Did Stephen Churchill get CJD from infected beef? And if so, was the country on the brink of a human epidemic? An outbreak of a similar illness offered a chilling clue. Early this century, a strange new disease appeared in the highlands of Papua New Guinea. It was called Kuru, which means to tremble with fear in the native language. Within years, it killed thousands of the foray people, but was unknown elsewhere in the world. In 1957, scientists traveled there to uncover the cause of this mysterious ailment. One of them, a young American pediatrician named Carlton Gaidushek, would one day win the Nobel Prize for this work.
They found a population that was dying from this disease, but primarily affecting children of both sexes equally and young adult women. The first symptoms were a little incoordination. They'll stagger a little bit when they walked. Little by little, over a period of months, this became sufficiently severe so that they were unable to walk unaided. In time, the people were unable even to stand and therefore became helpless. Most people with this disease died within 9 months.
Scientists investigated every possible cause of this illness, from malnutrition to genetic problems.
The answer turned out, in fact, to be very simple. These people were cannibals. clearly being transmitted from person to person by cannibalism.
Members of this tribe ate their dead relatives as an act of homage during funeral rites.
In the course of cannibalistic ritual feasting, the body was cut up into parts, and the men reserved the best parts for themselves, and the best parts were muscle. The remaining parts of the body, including brain and pancreas and liver and kidney and intestines, were eaten by the women and the children.
The disease was passed on as women and children ate the infected brains of their dead relatives. Many developed kuru, and when they died, they were ritually eaten, escalating this deadly epidemic. This tragic cycle was broken when cannibalism among the foray people ended in the 1960s. But the incubation period of this disease is so long that the last victims are still dying today.
The solution to the way kuru was passed from human to human, together with a number of laboratory experiments conducted over the years since, have, without any question, brought persuasive evidence to the idea that infection can be transmitted by feeding.
Cannibalism may be the cause of England's mad cow epidemic too. Cannibalism among cows and sheep. Scrapie is similar to mad cow disease, but it attacks sheep. It's called scrapie because infected sheep scrape their skin raw. Scrapie has existed in England for at least 200 years, but it has never crossed into cows. So how did cows get mad cow disease, known by its medical name as bovine spongiform encephalopathy, or BSE? Just as with kuru, food was the culprit. For several decades, cattle feed had included a cheap protein supplement made from the carcasses of other animals, including sheep and cows. BSE probably arose when sheep infected with scrapie or cows with BSE were turned into feed. The feed then infected other cows that ate it. And when those animals died, they were fed back to more cows, creating a rapidly escalating epidemic. It was a kind of cattle cannibalism, frighteningly reminiscent of kuru. And for 2 years after BSE was known, infected cattle were still allowed into England's food supply, raising fears that people might get BSE. To assess that risk, the British government called upon the scientific community.
Back in 1988, it was very difficult to be confident in one's recommendations. The amount of information that we had about spongiform encephalopathies was very small. We knew there was this disease in sheep, fairly widespread, called scrapie, which had been around for 100 to 200 years. It didn't seem to be causing a great deal of problems in the human population, so we based our conclusion that the chance of transmission to humans was remote on the long experience of scrapie.
As an extra precaution, the British government banned the practice of feeding cows and sheep back to each other. And cattle already showing signs of BSE were excluded from use in human food and destroyed. But these measures left people at risk. BSE has such a long incubation period that cattle that appeared healthy but were actually harboring the disease could still be sold and used in human food. And the government continued allowing many organs where BSE accumulates, including the brain and spinal cord, to be added to British meat products. Eventually, the British government called for the removal of these parts from all carcasses being slaughtered. But no one knows the extent to which infected cattle and contaminated beef products entered the food chain.
I've often reflected as to whether we wrote our report in too reassuring way, and in some way this dimmed the urgency which was necessary, and obviously one regrets that.
Critics also charged that the interests of industry were placed above public health.
You may wonder why we were not more alarmist in our report, but you must remember that at that time we were dealing with a very rare disease in cattle. A human disease is and has remained very rare. Most people had never heard of it. And if we had been too alarmist, we were in danger of upsetting the whole of the meat industry in Britain and elsewhere in Europe.
Outside of England, restrictive steps were taken. The European community banned the import of most British cattle, although packaged meat products were still allowed. The United States also imposed a ban on British cattle.
This country did a couple of things immediately. It first of all banned, as did most countries, all countries perhaps, the importation of living animals from Great Britain.
Going one step further than Europe, the United States banned all British beef products in 1989. Meanwhile, the Department of Agriculture began tracking down the several hundred British cattle that had been imported to the United States before the ban went into place.
We pulled the records for the 496 that were imported, and we had our field veterinarians trace those out, locate the farms where they were now residing, and then monitor those, check them every 6 months, where they would go out, talk to the owner, check for signs.
Those cattle never showed any symptoms of BSE, and most have been bought by the Department of Agriculture and slaughtered. But imported British cattle was only one issue. The question was brought up, could American cattle be harboring their own native strain of BSE? In 1985, in the Wisconsin town of Stetsonville, mink being raised for fur came down with a rare Spongiform Encephalopathy like mad cow disease. Almost overnight, they started showing classic symptoms of the disease, such as loss of coordination. They soon became listless and died within weeks. The disease wiped out thousands of mink. Richard Marsh, a veterinarian at the University of Wisconsin, was called in to figure out what had caused the epidemic. Marsh thought that something in the feed was spreading the disease. Now deceased, Marsh used to work in Paul Brown's lab at NIH.
Since mink are fed carcasses, they are voracious carnivores, the interest very quickly led to the idea that the carcasses were at the base of it and that they were getting infected through a sheep, specifically scrapie in sheep, and sheep carcasses were being fed to mink, so it made sense that sheep were the source of mink encephalopathy.
But Marsh began to suspect that on this farm, scrapie was not the cause.
No sheep had been fed, but downer cattle carcasses were fed. Downer cattle are called downer cattle because they lie down, because they're sick and they die. And many different diseases cause this picture in cattle. And the thought was, well, here you've got an outbreak of Spongiform Encephalopathy in mink that apparently had been exposed to downer cattle and not sheep, and maybe the cattle were the source of the outbreak.
The mink outbreak convinced Marsh that a low-level native strain of BSE exists in the United States, but was going undetected.
So the idea that BSE might exist in this country, either producing a disease that wasn't recognizable as BSE or existing as a silent infection, was one that we had to seriously consider.
And because of Dr. Marsh's theory, in 1993, the USDA started to do surveillance on downer cows. We've tried to approach it from a scientific standpoint and say, well, hey, if there is such a thing, let's look for it. To date, we've looked at over 6,100 brains and found no evidence of BSE.
So that's a pretty good piece of evidence that this is not the case in the U.S., but it's not proof.
Still, no cases of BSE have ever been found in the United States. But in England, throughout the 1990s, BSE was spreading fast, and not just in cows. British house cats started contracting the disease from beef and pet food. Like people, cats have never been susceptible to scrapie.
It was certainly concerning when domestic cats developed a Spongiform Encephalopathy, and we now know that is BSE that was acquired by these domestic cats, and now by quite a few wild cats kept in zoos and some other wild animals kept in zoological gardens. And so that really indicated that BSE had a quite different host range than scrapie. It was infecting different species that had not gone down with scrapie in the past. So that was certainly a cause for concern for me. Of course, that doesn't tell us that humans are going to be at any higher risk, but it does tell us that BSE is rather different than scrapie.
In many illnesses, including scrapie, biological differences between species prevent diseases that originate in one kind of animal from infecting another. This is known as the species barrier. For example, scrapie has existed for centuries, but no person has ever gotten scrapie from eating lamb. Unlike scrapie, though, BSE has proven highly transmissible, crossing into nearly every species exposed to it. For so many animals, the species barrier against this new disease was turning out to be weak, and there was no way to know whether it would be for people, too.
It really is quite an unpredictable phenomenon. By and large, we tend to think that animals that are more closely related in evolutionary terms are going to be easier to transmit the disease to. But it's not always like that at all. For instance, it can be extremely difficult to transmit the disease between mice and hamsters, which are quite closely related species. It seems to be relatively easy to transmit BSE to mice, which are quite distantly related species. So, there are really some very strange rules operating here.
When a Spongiform Encephalopathy crosses a species barrier, a frightening phenomenon can take place. As the disease passes among members of the new species, it can become stronger and more virulent, and its incubation time can shrink. Then, this new type of disease may be able to infect other species that were not previously susceptible.
We've certainly known since the mid-1970s that when scrapie infection is transmitted to and passaged within a new species, that strain characteristics and disease characteristics can change, and that a phenomenon analogous to mutation can occur, and as a result of that you can generate a strain with new properties, with new characteristics, with altered neuropathology, even with an altered host range.
Let's assume that BSE is the result of rendered, recycled scrapie in the food chain of cattle. There is not a shred of evidence to date that scrapie has ever caused CJD in humans. But scrapie passaged or going into cattle might change the host range of the infection, and therefore we cannot predict whether BSE would be or would not be infectious for humans.
We, of course, had no way of knowing at that time whether the BSE agent was exactly the same as the scrapie agent. Work since has shown that it does actually perform in a slightly different way to the scrapie agent.
This news was alarming. If BSE has a different host range from scrapie, could it infect humans, turning their brains to sponge? Answering that question was difficult because these strange diseases do not behave like conventional illnesses. Most infectious diseases are caused by bacteria or viruses, tiny microbes that can only be seen under the microscope. Bacteria and viruses contain genetic material, nucleic acid such as DNA. Nucleic acid is the essential ingredient of life and allows organisms to reproduce. Bacteria and viruses cause disease by spreading toxins or damaging their host. But spongiform encephalopathies seem to operate differently. Unlike bacteria and viruses, they provoke little or no immune response, signs that the body is fighting infection, such as antibodies. And they have another strange characteristic. In the 1960s, scientists found that radiation, which kills viruses and bacteria by destroying their genetic material, has little effect on spongiform encephalopathies. They appear to defy the rules of biology.
These agents are almost immortal. They resist alcohol, they resist boiling, they resist hospital detergents. We thought it would be interesting to see what would happen if we buried some of these agents. And so I ground up some scrapie brain and mixed it with soil, put it in a flower pot, and closed it in a cage, and used my own garden as a burial site, right here. And what we found was that a good deal of the infectivity remained in the soil after 3 years. We exposed it to temperatures that turned it to ash, and it did not entirely kill the agent. And so every known pathogen of man would have been destroyed by this process, and this was not.
So if these diseases don't behave like other pathogens, what are they? Scientist Pat Mers discovered a tantalizing clue in her New York laboratory. Using an electron microscope, which can magnify up to 100,000 times, she was looking for the infectious agent that causes scrapie. At that time, most believed the killer was a slow-acting virus. But her discovery pointed scientists in a new direction. She detected these hazy threads in the brains of animals infected with scrapie. They did not appear in the brains of healthy animals. Mers thought these fibers could be a sign of scrapie, CJD, and Kuru. But each of these strands contains millions of particles, far too small to be identified even with the electron microscope. A different technique was needed.
What we wanted to look for was what was unique about a sample from the diseased brain that was not in the normal brain. What component, what molecule would be in that diseased brain that was never in the normal brain? And so I did some experiments where I looked at proteins and separated these proteins using a gel to separate them by their size, and they give little different bands on the gel. And when I did that, I didn't really expect to find anything right off the bat. And after one of the experiments, I went into the darkroom to develop the film, and I was very surprised when I saw this. And I knew when I saw this gel that this had to be it. There's a fuzzy band that's here in the gel that's in each of the three